Hi guys, this is a quick review video on the vascular system, histology review. In particular, this is something that I wanted to make sure I had straight in my mind. Pretty much the different vessels that we're dealing with, because we obviously have artery, veins, capillaries, those are the big ones, but we have further sort of subcategories of each. And so it's useful to know what those are and the differences. And pretty much the first place we start in terms of talking about that are the different layers that vessels of the vascular system can have, right? And so this is all about moving, red and white blood cells, and then anything that needs to be delivered or waste removed from our blood system. So, anyways, we have in our vascular system we have layers that make up our vessels, and so not every vessel has all three layers. They generally do, but we'll mention which ones don't. But our vascular layers are connective tissue of type 1 and type 3 collagen, and they'll include elastic fibers. The innermost layer of our vascular layers is the tunica intima, and if I mispronounce something, I apologize, I'm learning this stuff myself as well. That will consist of first the endothelium on the very inside of the vessel, then the subendothelial layer, and then finally the tunica intima will end with the internal elastic lamina. After that, you have the tunica media, for middle, that's made up of smooth muscle, fibroelastic connective tissue, elastic lamellae, and then finally the external elastic lamina, EL. All right, and then you have tunica adventitia, two different color. This consists of fibroblasts, collagen, elastic fibers, and then smooth muscle, and then it will have the nervi vascularis, and the vasa vasorum should be additional tiny blood vessels meeting up with the vessel at the tunica adventitia. And so it's important to know that, of course, our capillaries, as we'll see in a second, aren't going to have this layer, and they're not going to have this layer, so they're not going to be dealing with vasa vasorum. They will contribute to the vasa vasorum network of smaller vessels connecting to a big vessel at the tunica adventitia, but the capillaries only have the tunica intima. Okay, so we have our vessels, we have arteries as our first category, capillaries, and then veins. So let's take a look at arteries. Arteries, we have our elastic arteries, our muscular arteries, our arterioles, and then our meta-arterioles, going from biggest to smallest. So our elastic arteries, they're going to have a tunica media with many elastic lamellae, so rings. They will have fenestrae, which I'm mispronouncing, I'm sure, but those are basically tiny windows or holes for diffusion. And then the tunica adventitia is going to have the vasa vasorum and the nervi vascularis, which is nerves that innervate the cells of the vessel, and then the vasa vasorum, again, is that network of smaller vessels that are going to feed into the bigger vessel. All right, and then it will have an elastic wall, which works to, it's facilitating blood movement. All right, so that's our elastic artery. Then we have our muscular arteries, and they're going to have a tunica media with a prominent IEL, which is our internal elastic lamina. They are most notable for having 3 to 40 layers of smooth muscle. So if you see large section of smooth muscle, that's going to be a good sign it's a muscular artery. Then we have our arterioles, which is going to have a relatively thin tunica intima, 1 to 2 layers of smooth muscle, and it's going to regulate flow to the capillaries. And before it gets to capillaries, we have our meta-arterioles, which are going to act as really sphincters. They do not have the tunica media or adventitia. Instead, they have individual smooth muscles, and they're acting as sphincters to allow blood to pass into the capillaries. Okay, and again, regulate flow with the precapillary sphincters, no tunica media or adventitia for the smaller vessel. Okay, then we have our capillaries. We have 3 different types. We have continuous, fenestrated, and discontinuous. Well, we know fenestrated means tiny holes. Discontinuous is going to be large gaps with little to no basal lamina. And all of our capillaries only have the tunica intima layer. Our continuous capillaries are going to have a continuous basal lamina. They're also going to have these cells on the outside called pericytes that are going to act as, they're going to be contractile, so they can squeeze on the capillaries to help with movement of blood through them. Okay, so not too bad. Then we have our veins. The veins, we have 3 different types we're looking at. We have postcapillary, postcapillary venules, muscular venules, and large, medium and large veins. Our postcapillary venules are going to be like our capillaries. They have the tunica intima only, and they're importantly the site of leukocyte migration and inflammatory response. So our leukocytes will leave vessels at our veins, our postcapillary venules. And then we also have, then the next one is our muscular venules. You have all 3 layers again, and we're dealing with 1 to 2 layers of smooth muscle. So similar to the arterioles, the muscular venules. And then we have the medium large veins, which has all 3. The tunica adventitia is predominant in terms of size over the other ones. Okay, and then a couple last things to mention. We have AV shunts, which stands for arteriovenous shunts, which is direct arterial to venule connection to bypass capillary beds. And then portal systems, important to know as well. It's 2 capillary beds in series. If it's an arterial portal system, that means that the efferent arteriole is connecting the capillaries. An example, this is the renal cortex. And then if it's a venous portal system, it is a vein connects the 2 capillary beds, and it is hepatic. An example is hepatic portal system, and the pituitary gland has a portal system as well. All right, so that's it for the quick review. This was covering lecture 77 of histology, and we'll take a look at the lab 6, which deals with this content in just a bit. Good job.